Are you guys ready?
When you average it with 3.
Ready?
What are we praying
For what?
Immune system?
Oh, go for it.
Oh, God, yeah.
You ready?
Give it to me.
Okay, so, the immune system is interaction between antigens and antibodies and all the other things within that. So the first thing you have to talk about is antigens. Antigens are anything within the body that is considered not self, and that could be like proteins or chemicals and stuff like that. And so antibodies are proteins that recognize antigens. And this is kind of what an antigen looks like, or excuse me, an antibody. It's made of two, these are like exactly the same. They're mirror images of each other. And it's made of two chains, a light chain, which is like smaller, and then a heavy chain. Then there's, within these chains, there are conserved regions and variable regions. So these boxes here are what define the variable regions. And then these lines are the highly conserved regions.
And what does that mean?
So every antibody has this shape. And that's because of these conserved regions. And then the variable regions are what recognize different antigens. And that's what makes it really useful, because these variable regions will bind to whatever it recognizes on the antigen. So antibodies bind to antigens, and the way that they get rid of it is they form these aggregates, like just this big mass of antigens, because it'll stick to, the antibody will stick to antigen, other antibodies will stick to other antibodies, and it just makes this big mass. And so whenever the body sees these big aggregates, phagocytes,
Phagocytose.
yeah, they phagocytose them and get rid of them. So these variable regions are defined by clonal selection, and there's a locus within the genome called the VDJ locus, and these three genes, each one is made up of a bunch of exons. So each one of these is just a whole lot of exons. Each one has a different amount of the exons within. And so there's this enzyme called VDJ recombinase, and it randomly picks out one of these exons and preserves it. Or rather, it keeps these and cuts everything out. And then in the end, you only have these three exons. And that's what makes this variable region, are these three exons.
And so there's a lot of variable regions then? There must be a lot of different types?
No, there's only, so this one has 100 or something variable regions, or exons.
But all the different combinations?
Yeah, there's a whole lot of combinations that you can make.
Okay.
Because it only chooses one, and then it only chooses one from all three of them.
Okay.
So that, multiplicatively, just you have an exponential amount of different combinations. And each B cell, or each immune cell, is specific to one of these combinations.
Okay.
Or, in other words, you'll have one B cell, and the VDJ recombinase in that one B cell will remove all this DNA such that it has this specific VDJ combination in that one cell. And let's just say that VDJ combination is VDJ1, right? So then there's another B cell that's forming at the same time, and it's going to have VDJ combination 2, right? And they're two separate cells, but they're both specific to different things based on those combinations. So yeah, let's go. So then this is the pathway of how an immune response is with a B cell, and it the way the immune response is it's an interaction between B and T cells, but I'll talk about that later. So what happens is B cells are actually coated in antibodies. So like the entire surface is covered in these antibodies, and whenever it sees this antigen, so it could be like anything, it could be something on the surface of another cell, it could be something floating around in the extracellular matrix, it could be anything. Once it recognizes it, like the variable region touches, it endocytoses it, and it like chews it up and like breaks it up, and then this molecule called MHC class 3,
3 or 2?
2, sorry. It takes a piece of that antigen and presents it. So now this becomes an antigen presenting cell, which is the next APC. And so T cells localize to antigen presenting cells through the interaction of MHC2 and T cell receptor, or TRC. And what happens is there's a reaction between these two, and it induces the B cell to divide into 2 different types of cells. One of them is an effector cell, and the other is a memory cell. What the memory cell does is it is exactly what it sounds like. It stays in the lymphocytes, and it's actually the hallmark of the adaptive immune system. And
there's also another
what that does is it
stays
stays in the lymphatic system, so that when the body sees this antigen again, it can react much faster than it did when it saw it the first time. So that's the importance of the memory cell. And then what the effector cell does is it just makes a lot of antibodies, and it just spews out all these antibodies and goes all over the extracellular matrix and forms those aggregates, and then the aggregates get phagocytosed, and that's the immune response.
Awesome.